Okay, next we have Natasha Latysheva with us. She's a research engineer at Google DeepMind, a senior research engineer in the Genomics Initiative. Her work focuses on integrating deep learning with molecular biology and genomics, particularly in understanding regulatory DNA and predicting variant effects. She holds a PhD in computational biology from Cambridge and a biochemistry bachelor's from St. Andrews. Before joining DeepMind, she worked in data science and machine learning roles in gaming and natural language processing. Over to Natasha.
Thank you so much. Yes, so I am indeed a research engineer at DeepMind, working in the genomics team. I've been there about 4 years now. We're part of the broader science team in DeepMind, so that's the team doing all the biology and physics and maths and so on, some of which you've heard about today. So yeah, really excited to share my field with you. This is the outline of the talk. So I thought I'd spend a decent amount of time first introducing the field. So what is genomics? Why are we trying to do machine learning in genomics? What kinds of applications open up if we were to do a really good job at modeling genomics? And then, because this is a machine learning audience, I'll spend quite a lot of time going into more depth about the current state of the art in DNA-based deep learning models. I'll talk about Informa, which is a DeepMind model, and also Borzoi and CompuPNet. And I'll touch on DNA language models a bit. I'll talk about nucleotide transformer, although now I know that there's an InstaDeep researcher in the front row. I'm feeling a bit self-conscious there, but I'll do a decent job, hopefully. And then I'll finish off with future directions. What are the main challenges in the field? Where do I see this field going and developing? And then finally, a conclusion where I'll end with some motivational messages, which will make everyone quit their fields to join genomics, hopefully. So yeah, so we can get started. Introduction. So I guess the first question is, what is genomics and what is a genome?
It's like blocking
Oh yeah. Thank you. Right, so a genome is simply all of the genetic material in an organism. It's the totality of the DNA in the cell, mostly in the nucleus, some in the mitochondria. And genomics is the study of the genome. So what's in it? How does the genome work? Where are the genes? How are they controlled? What makes gene activity go up and down across different conditions and cell types? And I guess you could contrast genomics with genetics. Often in genetics, people are studying one gene or maybe a few genes. In genomics, we tend to zoom out a bit and consider the totality of the genome, maybe genomes across individuals or even across species. And yeah, essentially the genome is very long and complex. So in the human genome, we have 3.2 billion letters or bases. And even though we've had the human genome sequence since 2003, there's basically just a huge amount of unanswered questions about it. How do all of these letters come together to form a cell, which then comes together to make tissues and organs and then complex organisms? And there's a famous quote by Eric Lander, who's a famous biologist. Genome bought the book, hard to read. This was said about 20 years ago, and I would say that we're still in the beginning stages of learning how to read this super long, super complicated book. But there's hope. There's hope in machine learning, and there's reasons to think that deep learning will be very useful in the DNA domain. Firstly, because it seems to be really good everywhere else, basically, every problem that you throw them at. Of course, yeah, machine learning is really good in the text domain, and it has been for a while, actually, even before the recent LLM boom. And also images. I'm sure a lot of people in this room have started their machine learning journey doing image classification, classifying MNIST digits. I hope they've solved that problem by now. But yeah, the field has come a long way since then, and we can now do really cool image generation. And fun fact, if you add the word cyberpunk to your prompts, you get very cool images out of that. And you will be subjected to many more cyberpunk images in this talk, so I apologize in advance. So yeah, deep learning, really good at text, really good at images. Video generation has really come a long way as well. Video games, this is a few years old now. This is AlphaStar playing StarCraft 2. Music generation, I would say also really good, a bit of a lag maybe, and then most relevant to this talk is protein structure. I think it's fair to say this has been the most successful deep learning model in biology, and it's also in molecular biology, so proteins and DNA are quite closely tied together. So, what could we do if we had a really good advanced understanding of genomics? There's a few immediate application areas that pop up. The first and I think most obvious is understanding disease mechanisms. I think people are often surprised by how poorly we actually understand how a lot of diseases work, especially rare diseases. We have good statistical methods for linking genetic variants with the presence of a disease or the severity of the disease, namely genome-wide association studies, or GWAS. But this often doesn't really tell you much about how the disease happens. So it'd be really nice if we had models to bridge that gap. And this ties in quite nicely to the next point. If you don't really understand how a disease works on a molecular level, it's really hard to then fix it, to design drugs to fix it. So if we do have that knowledge, this informs drug discovery. Better understanding of genomics would really also advance cell engineering, for example, transforming cells of one type into another type, such as taking a skin cell, transforming that into a stem cell, and then into a neuron, and using that to treat spinal cord injuries. And then on the preventative side, do I have a cursor? Oh, no. And then on the preventative side, it'd be nice to improve disease risk prediction. If we can stratify the population into low-risk groups and high-risk groups, then we can do preventative treatments on high-risk groups, for example. And then disease aside, understanding population variations, another application area. What is it about your genome that makes you you? This feeds into consumer genomics as well, things like 23andMe. Improving gene therapy. So gene therapy is already here. It's quite futuristic, but we already do it. It's quite successful. The basic premise there is if you have a mutation in a patient that sort of affects the normal function of a gene, can we introduce functional copies of the gene into the right cell types of that patient to rescue the function? And I think the sky's the limit with engineering metabolic pathways. Here I mean things like improving pest resistance in crops, improving drought resistance in crops, engineering bacteria to degrade contaminants in the environment, optimizing algae for carbon capture, these kinds of things. And that was a lot of applications, but I think more on the academic side, I think genomics also offers a really rich approach to understanding some of the more fundamental questions we have as people, basically, like what is life? What is the basis of life? How do cells work? How has life evolved over the last 3.5 billion years? Really fundamental questions there. And then if we do understand evolution, then perhaps we can predict evolution. If we know the mutation trajectory of a virus, can we start to predict the probability of new mutations arising? Can we design drugs against those mutations before they even occur? Can we do the same thing for cancer cell evolution? So each of these points is a massive field in itself, but I think the overall take-home message here is on the practical side, if we were to do a really good job with genomics models, this would really revolutionize preventative healthcare. And if a patient already has an issue, then we could design more powerful and more personalized medicine to take care of that issue. And on the academic side, we can understand more the basis of biology and how it has evolved. So given all these opportunities, why haven't we done it yet? So it turns out this is a really hard problem. I think primarily for reason one, most of all. So this is discerning between meaningful biological signal and noise, and there's a massive amount of noise in the types of problems I'll explain in this talk. But yeah, to summarize, DNA sequences are super long, even for modern machine learning algorithms. A lot of them are not useful to a problem. If you're trying to predict the expression of a gene over here, most of the letters are not going to be relevant, and maybe like 10 are hundreds of thousands of bases away. The individual tokens are also not really informative. If you think about it, you have 4 bases. There's just not a lot of information there. Tokenization is tricky. So yeah, there's just a lot of biological noise. There's also technical noise. There's sequencing errors. There's, if models train on labeled data like RNA-seq and ChIP-seq, the models inherit the biases of these assays. There's also batch effects where different labs performing the same exact assay or experiment on the same cell types will come out with different results. So yeah, there's just a lot of biological noise and technical noise, and it's really the ultimate needle in the haystack problem. And one thing that has become clear over the last few years is that a lot of the models that we do train in genomics struggle with generalization across different biological contexts. And by context, I mean things like different cell types, or the same cell type but in a different developmental stage. Maybe you have a good model of skin cell gene expression, but you trained it in embryo, it doesn't generalize to adults. And also generalization across species is really difficult. One surprising thing is models can struggle to generalize across individuals of the same species as well. So they kind of have overfitted so much to the reference genome where most of the data is available. So this is a huge issue that people are working on. And finally, we do lack ground truth for many genomic problems. Yes, it's true that DNA sequencing has become cheaper and we have our own kind of Moore's law. A lot of the data is not really easy to use or super relevant. Supervised models still kind of reign supreme, and we just don't have great labeled data for a lot of contexts. Cool, so yeah, if there's no questions on this introduction, I can dive into how these models work. Awesome. Yeah, I did a bit of this intro already, but I think it's fair to say DNA is the basis of all life as we know it. Maybe some alien species will object to this slide, but for now, this is the case. And the DNA in the cell contains all of the information that you need to grow into an entire organism. So the DNA also encodes how all cells will respond to stimuli, how they grow, how they divide, how they transform into the thousands of different cell types that you have in the body. And ultimately, they contain the key for how diseases work. And I think it's safe to say that understanding DNA is foundational. It's a root node problem. If we were to solve it, we basically solve a lot of other problems that stem from it. So we want to build models that understand the relationship between DNA and the next step. So between DNA and molecular biology, different properties of DNA that then filter into proteins and into tissues and diseases and so on. And I think understanding DNA is really prerequisite for any of the applications on the previous slides. So a bit of a recap of information flow in biology. There's this concept called the central dogma, which basically states that DNA makes RNA makes protein. So yeah, you see some DNA there and a gene where that arrow is. This then gets basically copied by some cellular machinery into transcripts or mRNA. And the mRNA is the second stage there. That will then get translated into protein. So there's no proteins without mRNA and without DNA. I mean, there are some exceptions. You can go in the other direction, like some viruses will reverse transcribe into DNA. And sometimes you have protein-to-protein information flow in prion diseases, but mostly this is how information flow happens. Right, and so just to give you a scale of GWAS, this is actually even a few years old, but if you were to log on to the EBI GWAS catalog, you'll see a diagram like what's on the right. So this is a single chromosome, and you can mouse over all the dots, and this is basically a statistically significant association between a variant on that chromosome and a disease or a trait like height or BMI. So we have these maps now where we know variants are associated with something important, and we have many, many more of these hits over time. But a lot of the time, that's where our knowledge ends. We don't know how these variants filter down to actually have effects at the mRNA level, at the protein level, which cell types are affected. We just know that the statistical association is there. So if you have an A at a certain position on chromosome 17, position 2 million, that means you're more likely to be healthy. And if you have a T or a T allele, that means you're more likely to be diseased. So we have these kinds of associations, and we really want deep learning to bridge this gap in understanding what they're actually doing on a molecular level. This is a bit of a draft diagram, but I tried to put together the types of DNA and RNA modalities that different deep learning models have been covering over the years. I think this field kind of started, does that say 2013, roundabouts? So we had some initial models there, like DeepC, Basenji, and so on. And I guess you can kind of cluster models in this space into two types. Some of them are quite specialist models that just cover one thing. Like Saluki on the bottom right here just covers RNA stability, and it does a really good job of that. So given an RNA sequence, how stable is that transcript? What's its half-life? How quickly is it going to degrade? This varies quite a lot. And other models take a more multimodal approach, which I quite like, actually, because all these processes are connected, but of course it's a much more challenging modeling problem. So I think it's hard to understand what those words on the left mean unless you're already working in functional genomics, so I apologize for the jargon. I'm just going to cover the four most important ones. The first is gene expression. So this is basically, given a gene in the genome, how often is it being read? How many copies of that gene are being created? And this correlates quite well with how many copies of the protein are going to be floating around the cell. When I say assay, I mean an experiment in the lab done to measure something. So our best assays for measuring gene expression are RNA-seq and CAGE. And a lot of different models try and predict gene expression. This probably, if you had to model one thing, you would model gene expression because genes are really important. Another concept that will recur is chromatin accessibility. So the idea here is that, when you see pictures of DNA, you see like a nice double helix. This is not really realistic. Most of the time, DNA is quite tightly packed away. It gets wrapped around some proteins called histones. That then gets packed into bundles, which gets packed more, and it can become quite condensed. But this concept of accessibility is quite important because basically for DNA to be active, it has to be open. Like if you have a gene, but it's packed away, it's not going to be read by any of the machinery. It has to be unraveled first. So third concept is transcription factor binding. So DNA is full of many, many binding sites. Different proteins will bind to different binding sites. A protein will have a specific preference for a certain sequence. It's kind of wiggling around the cell, sometimes binding here, sometimes there, binding more strongly where the motif or the sequence of the DNA matches its physical shape. And transcription factors are a class of proteins that are very important for gene expression. There's different types that activate gene expression. There's other types that repress it. The top view on the left is kind of a linear view. So RNA polymerase is the protein that will actually start transcribing a gene, so starting gene expression, but it's dependent on transcription factors binding to the promoter at least, and then also maybe enhancer sequences. So that's sort of the linear view. DNA is not normally like that nice. Usually you get this loop formation where the bits that promote gene expression, so enhancer sequences kind of fold over onto the start of a gene, and that actually allows, kind of a complex of proteins to come together to then actually express the gene. And then finally, sorry for all the biology, but I did biochemistry, so I'm compelled to put these diagrams in. Finally, we have histone modifications. So I told you that DNA is kind of wrapped around these proteins, and then there's these little tiny tails on these proteins that get modified, chemically modified with different tags. This turns out to be really important for the activity of genes, and they're really good markers of like repressed regions of DNA or active regions of DNA. So now if we were to go back to this slide, you can see that lots of different people, different research teams have made all these different models to model these different modalities. And you can see some of them there, expression and chromatin accessibility and so on. Cool. this is just a reminder that the cell is just complete chaos, basically. It's really busy, it's a very crowded place. Lots of these molecules are wiggling back and forth. We have these nice diagrams about what's going on, but just a reminder that, like, yeah, it's really busy and crowded. Right, so this is a model from us, from DeepMind, 2021 now, which is a while ago. Wonder what we've been up to since then. I can't tell you, but maybe dinosaurs, who knows. And so the task of this model was to predict gene expression from DNA sequence. So basically, I wish I had a pointer. But basically, we train like a hybrid convolutional and transformer model. We had quite a wide receptive field, so we were able to model longer DNA sequences than ever before. But basically, the convolutions and through pooling, we sort of shrink down the DNA sequence. And then to the transformer blocks, we send a much shorter, lower resolution sequence. Because attention scales very poorly, like you kind of have to do this aggregation bit first. And eventually we're able to predict these genomic tracks. And by genomic track, I mean like a readout over the DNA sequence, such as expression or chromatin accessibility or these other track-like concepts I introduced earlier. Right, and then we did a lot of evaluations and basically, yeah, at the time and until a few months ago, this was state of the art at various tasks like predicting gene expression or predicting the effects of variants that change expression and so on. And we also did some nice interpretation work, which I'll dive into in a second. Cool. So this is the training data for the model for Informer. So one thing to say is that we're incredibly dependent on experimental labs for generating data, basically. And in genomics, there's this culture of massive consortia of like hundreds, sometimes thousands of people generating this data, and we're extremely grateful for them because basically, yeah, any ML work wouldn't be able to get off the ground without them. This data is from ENCODE and Phantom 5. So we trained Informer on thousands of different tracks simultaneously, so this is a massive multitask model. We had data from humans and from mouse. CAGE is the expression data. We weren't using RNA-seq yet at the time. And then DNase is the chromatin accessibility data, and then there's some histone modification data as well. So I guess the input tensors and output tensors are, input is DNA sequence, and output is these 6,000, 7,000 tracks. Oh, actually, one thing I haven't said yet. So, if you're modeling DNA and you want to predict expression, expression varies quite a lot between cell types, and we want the model to capture that. So we don't want some sort of average expression over all cells. Given a DNA sequence, we want to know, like, is this gene expressed in brain and all the different subparts of brain? Is it, you know, expressed in kidney cortex or tibial nerve, like really specific cell types. That's why there's so many thousands of tracks. It's because you have CAGE tracks for like many different cell types. Yeah, and basically if you have a look at the model predictions, they look pretty good. The green is the experimental values, blue is the predicted values, orange is the sad predicted values of the predecessor model, which we're forced to include. And basically, yeah, we get quite high correlation with actual gene expression, 85%. And yeah, this is just showing visually some examples that the peaks are where you would expect based on experimental data, the gaps are where you'd expect. Yeah, and no major problems really. This is the predecessor model, Basenji. This is just to say that their input window was a lot smaller, 40 kb, 40,000 DNA bases. And also this was before transformers, basically, so they just had a pure convolutional model. I think it does pretty well, though, yeah, for the time, for sure. And we managed to expand the view quite considerably to 400 kb. And by adding these transformers, we can model some interesting biology. So you might have seen this diagram from a machine translation example where I think it's translating English to French. and you have these different word tokens kind of attending to the most relevant tokens in the other language. The biological form of this is what I mentioned before. You could have a gene over here, but actually there's a regulatory region all the way over here that controls the gene, and in reality the DNA kind of bends around to put these regions into contact. And if you inspect the attention matrices on this model, you can see that you get this pattern. Basically, you get the right regions tending to each other, which is really cool. This is an important point, actually. Some people think, and I think, my team thinks, that it doesn't really matter how well you can recapitulate experimental data, like how well do you match these peaks and troughs. What's actually important is downstream tasks of predicting what is the effect of mutations, so predicting variant effects. Just to make that a bit more concrete, so as I said, we train on the reference genome. So imagine that's the top right ATCG-TA. And we make certain predictions off the basis of that DNA sequence. Now, what if you changed a single letter in that DNA sequence? So you're introducing an alternative allele. So you put a G where there was a C before. What is the difference? Can you predict the difference? We know the difference experimentally, and sometimes it can be massive. So a single letter change can absolutely change the game of how that DNA is behaving. But the question is, how well can the model predict that difference? So this is a really hard problem. But at the time, we were state of the art at predicting the effects of expression variants. These are called EQTLs, expression quantitative trait loci. And these are variants that through GWAS we know change the expression of some gene up or down. One thing that's interesting, though, is that the model really kind of falls off a cliff if you ask it to predict variants that are further and further away from the gene, so further and away from the transcription start site. If it's within 3,000, we do a really good job, actually, but the further away you go, the model struggles with distal information, essentially. And that's been a recurring theme of... I was just saying that we really care about using these models to predict the effects of mutations. Informa did a pretty good job, but if the mutation's far away, not even that far away really, like over 3,000 bases away from the gene, then the models really struggle. We did some interpretation work here. So the question we asked is, so the genes are kind of those lines at the top, and we predict expression peaks in the green. But the question is, when the model predicts a peak, saying that that gene is expressed in that tissue, what information is it actually using? And is that information tissue specific? So yeah, I don't need to explain everything, but basically we have labeled data sets where we know regulatory regions are, and we want to check that the model is attending to them appropriately. And we know these regulatory regions are cell specific, and yeah, basically we're able to show with some techniques like input gradients and mutagenesis that the model looks at the right things. So just a bit of a sanity check. Then there was this analysis paper that came out last year, which is actually one of my favorite papers in a while. This is from an external lab, Gagne lab in Germany. And there's a figure that showed that, again, the model really struggles to pick up on distant regulatory information. So if you look at the left-hand side, FOLCO, so there's these regulatory regions enhancers, which enhance the expression of genes. And the further away they go, it is a slightly weaker effect that's in the red, but not much. So you have some really strong enhancers far away. But as far as the model is concerned, it basically discounts enhancers the more away that it goes. So really, these two lines should have the same slope, but you can see the model is blind to distant information. Interestingly, we just hired this guy that did this analysis, and we kind of said, like, look, you've showed us a problem with the model, like, now come fix it for us. Also, yeah, doing some ablations. So different models in the field use different input lengths on the DNA, but how important is that actually? This plot showed that it basically saturates at a certain point, and you don't gain that much more information. And that's because a lot of regulatory information kind of lies within 50,000 bases of genes anyway. So just to summarize this model, so this model is called Informa, and Yeah, I think it captures quite well DNA variation that acts as a common mechanism. It's represented quite well in the data. And if the regulatory mechanism is strong, but if the sequence variation is less common or it's a weaker mechanism, yeah, it doesn't do super well. So I guess the key machine learning challenge is how do we encourage a model to pay attention to patterns that are less common in the training data, but which are still biologically important. So that's all I had to say about Informa. This one is not a DeepMind model, I guess still an alphabet model. This is from David Kelly's group in Calico, just published in Nature Genetics, I think, this month. And essentially, this is currently the state of the art of DNA deep learning. I think I'm happy to say that. They did a really great job. And basically, people for a long time thought that modeling RNA-seq data was too challenging. So that's the examples on the left. So this is what RNA-seq data looks like. You might have noticed before, we were modeling gene expression, but it was just a single peak where a gene started. RNA-seq data is more complex. There's peaks at every exon, basically. And the reason this is so challenging is in order for a model to predict RNA-seq well from DNA sequence, it actually has to understand like a mind-blowing amount of things. So first is gene transcription and regulation. Remember DNA sequence is just like ACGT, ACGT. It looks like nothing, but the model has to understand where the genes are, where are the promoter sequences that govern that gene's expression, where are the enhancers, where are the silencing elements, how do all these elements influence expression. It then also has to understand all the steps that happen to DNA after transcription. So I'll talk a bit about introns and exons just now. Yeah, basically, we have the nucleus on the left, and we have the outside of the nucleus, the cytoplasm on the right. So I've said that DNA gets transcribed into RNA, but then there's a process of RNA maturation, where the gene kind of gets shuffled together a bit, like certain bits get cut out. One end gets capped for stability, another end gets a tail added for stability, and then it's exported into the nucleus. And basically, in order to predict these RNA-seq wiggles well, the model has to have an implicit understanding of all of these processes. So I think the field was just a bit scared for years to approach the problem. And yeah, we're quite happy that David Kelly's group in Calico managed to do this quite well. One concept that I think is quite fascinating is how much diversity these processes introduce into the cell. So you have one gene, but with alternative splicing, where different exons get kind of concatenated together, some get skipped, it can actually produce many, many transcripts from that one gene. And sort of a sister process is alternative polyadenylation, where this tail of A's can get added to different locations and produce diversity that way. So I always think it's really interesting that evolution has sort of innovated here and produced a way of, just introducing diversity from the same sequence. You could make new genes, for example, if you need to evolve a new function, or you could make different versions of the same gene. And these are very important processes for disease. So we know that about 15 to 20% of human diseases are caused by errors of the splicing process on the left-hand side. So basically Borzoi managed to do it. They predict RNA-seq really rather well. This is their architecture. It's actually very similar to Informa. They kind of did a UNet step as well. They use the model to predict the effects of variants. Because RNA-seq is sort of many, many spikes over the gene, they can make some really nice plots showing that if a mutation reduces the expression of a gene, you actually see that across many peaks, that it's sort of a lower number. And as people always do in these papers, they do some interpretation to check the models picking out the right subsequences of DNA as it makes those predictions. Actually they had an author on the paper, Johannes, who loves model interpretation, as I do, and they have really gorgeous figures. This one is an attention matrix from one of the transformer blocks, and it's basically showing that if you overlay that with the known genes in the region of that DNA sequence, you can find that the attention blocks recapitulate gene boundaries really well. So this is saying, as the model makes its predictions, within a gene, all those tokens are looking at each other quite a lot. And you also see these vertical bands saying that in the region, all genes are looking at the start sites of all other genes. So I thought that was quite fascinating. And then some sanity checks, like if you scramble a region that you know is important, like a promoter, does that lead to the gene being predicted less? And indeed, it does. So I appreciated that. So that's all I had to say about Borzoi. Finally, chromBPNet is not primarily a gene expression model. This is more of a specialist genomics model that just wanted to predict chromatin accessibility, which, if you recall, is how tightly bound DNA is versus free and accessible to do its business. And I just wanted to, I think it's really fascinating, the innovation that they did here. So basically, what they're saying is, yes, we can predict these peaks like other models, but in the non-peak regions, how much of that is actually noise due to the bias of the enzyme that's used to generate the experimental data? And it turns out a lot. So this is Anshul Kandoji's work from Stanford. And basically, the enzyme used to generate this data is called TN5. And it will cut DNA where it's accessible, which is then, you can see in the readout. But it still has a preference for the types of DNA sequences it cuts. So it'll preferentially cut some sequences more than others. So what they did is they kind of trained two submodels, one to predict the peaks and one to predict the noise. And it worked really well in a kind of counterintuitive way. So yeah, basically the results look worse, but are not. So with the uncorrected model predicts the peaks much better in a way, but a lot of that is noise essentially. And if you remember what I said earlier, in a way it doesn't really matter how well these models do at predicting these tracks. What we really care about is downstream applications. And they did a lot of work, loads of benchmarks. Actually, it's a massive benchmark paper. They showed that the model gets improved performance on variants that are known to affect chromatin accessibility. So they're outperforming models like Informa and other models at this task. So I thought this is a really nice decomposition. So given a DNA sequence, predict how open it is, but at the same time, predict how much noise was introduced by the experiment. Cool, so I'll go briefly over this. It's not really my area of expertise, but DNA language models are becoming a hot thing. LLMs have, of course, been a complete revolution, and they're trained on these massive text data sets. Well, we have a lot of DNA data sets. DNA sequencing is becoming really cheap. RNA sequencing is becoming cheap. Why don't we do the same thing and in an unsupervised way train DNA language models to do their thing, and that way we can solve biology, essentially. So more and more people have done this. There have been some fantastic models, things like nucleotide transformer, DNA BERT, EVO, hyena DNA. Here I'll just talk about nucleotide transformer. And essentially, yeah, so either with next token prediction or mass token prediction, you train them exactly the same way. And you can show that their embeddings make sense. So if you look at a typical gene structure diagram over there, so we have a gene, but it's composed of a promoter region that sort of will start transcription. It has introns and exons. The introns are then spliced out. If you then look at the embeddings that the model has learned, you can see that it kind of corresponds quite well-ish to these different regions, showing that the model has indeed learned to distinguish between different types of functional DNA sequences. But then again, coming back to why does it matter? How well does it do at variant effect prediction? Well, it's actually not bad. It's not as good as supervised models, but I think it's quite cool. So they do a good job at predicting whether a variant will increase or decrease expression, whether a specific mutation will change if the DNA is methylated. This is like an epigenetic modification. And it also does a good job distinguishing pathogenic mutations versus matched non-pathogenic mutations. So it's quite cool. So I am really excited by the DNA language model concept. I think it is inherently more scalable. But there have been challenges, essentially. So a lot of the signal is hidden away. So we again have the signal versus noise problem. Much of the DNA sequence is not really that interesting or functional for many problems. The tokenization is not easy. Single tokens not informative. N-grams don't work super well. Bipair encoding sort of works, but not amazingly well. What are the scaling laws? We don't really know those yet. How far will this go, given that much of the genome is functionally not super relevant, especially. And one thing that actually I think is a very strong point is that genomics doesn't really have such a culture of standardized benchmarks that everybody can look at and say, yes, this is the final word on how good this model is. I think models like AlphaFold benefited massively from that because you can overlay the structure and see like, oh yes, this is similar to the ground truth. Genomics is, yeah, I hope it can get there. It is more complex in a way, but it can be hard to say whether a model's actually good or not. And then there's very strong competition to DNA language models from supervised models like Informer and Borzo and CronBPNet. So I would say those are kind of the challenges of DNA language models. Right, so those are the models I wanted to cover and some future directions here. So the first is data. There's really a bizarre like abundance and shortage of data. So the human genome is actually not that big. Yes, we have 3.2 billion letters, but we only have 22,000 protein coding genes. And if you think about train validation, test splits, and how long the input sequences are, you can end up with really not that many training examples. Some of the DNA sequences are repetitive regions or are like ancient viruses that are, transposons that are in the genome that are not super relevant anymore. And we don't really use individual genome data that well yet. There's very little experimental data from other species, other than like mouse and some other organisms, and it's not really clear how to integrate these into our models. In a perfect world, if we had high throughput CRISPR, if we could do really high throughput experiments, if you mutate this, this happens, that would really change the game, but the technology is not really there yet, and we can't edit humans. And the animal models and the cell line models that we have are fine, but they have their own weaknesses. So essentially, yeah, we're a bit dependent on the data and a little bit stuck here. As I mentioned before, the benchmarking culture is not really mature yet. This is the CASP example for AlphaFold. And then one thing that I'd really like to see in the next few years is more people combining these different phases of molecular biology, so combining DNA with RNA and protein into a single understanding of molecular biology. I know you have to kind of divide and conquer, but I'd like to see a bit more cohesion. And then finally, we're just still really quite early in the game in the AI for genomics space. So first human genome was sequenced in 2003. The first end-to-end human genome was actually 2 years ago. And although the cost of sequencing has gone down massively, we're really not sure how to leverage a lot of this data. One final thing, one final difficulty. So I told you about this linear view of the genome. It's actually like a bowl of spaghetti in the cell where the different colors are chromosomes and they kind of congregate into chromosome territories. The models we have do not really do spatial or time components like at all. Some of it is kind of bundled up in the embeddings, but it's not explicit. So I'd like to see more of that as well. So some technologies to keep an eye on. Single-cell genomics has been around a while. Multimodal genomics, basically before, to perform these experiments, you would have to kill the cell. So if you want to measure the gene expression of a cell, you can, but it's gone. Now you can at least measure the expression and the chromatin accessibility in that cell. So you can do 2 modalities, 2 assays. And I think that will just keep increasing. Spatial transcriptomics, I find super sci-fi. And this is mapping gene expression in 3D space. So yeah, I guess classically gene expression, you kind of blend a tissue together and then do the experiments, but there's a lot of spatial heterogeneity, and I'm really happy that people are starting to work on this and make these high-resolution atlases of different tissues. Large-scale CRISPR screens are the absolute dream for linking non-coding variants to their effects on genes, but yeah, that's for the experimentalists to figure out, I think. And then, yeah, just finally, some genomics projects to keep an eye on. All of Us just came out. This is a big project in the US, releasing loads of different genomes. UK Biobank is lots of genomes together with paired decades of health records. So obviously lots of people are working on this, but it's a gold mine, essentially, and we're trying to mine it for insight. And the Human Cell Atlas is a complete map of all human cell types and single cell sequencing across all sorts of different conditions. Great. Okay, so conclusion, a bit of a recap of this entire talk. Most of the deep learning models in this space focus on building models of DNA sequences and predicting the molecular function of that sequence. And usually, yeah, they're just trained to predict the experimental readouts, kind of track like peaks from the DNA sequence. And there's different types of experiments. I showed you ones that capture gene expression, ones that capture DNA packing, ones that capture proteins binding to DNA. And a lot of these models do a great job of predicting the effects of variants, but yeah, there's some difficulties like capturing distal information. And yeah, the field is fairly reliant on these experimental assays. So main challenges, just to summarize, we've got a needle in the haystack problem here. And yeah, the data is often noisy for biological reasons, for technical reasons, and frankly is kind of insufficient at the moment. It's hard to leverage single cell data and multi-species data. You think that you could just throw it at the models and they would improve, but it's really tricky. And the models we have can struggle to generalize to new biology. I think as benchmarking improves, a lot of these problems will be improved as well. Hopefully in the future, we'll see better integration of biology at all these different levels. And hopefully we'll also start to incorporate notions of space and time in the models. Yeah, so this slide is for students or other people interested in the field. I think there's so many different ways to get involved in the space if you liked what you see. So first, if you have more of a biology or medical background, you can help interpret the models, you can help understand the data, the experiments. It's a really valuable asset to any team. And these teams tend to be extremely multidisciplinary as well. So whether you're on the biology side or you're more on the AI, ML engineering side, where you're kind of building the models, analyzing them, both are extremely valuable. For myself, I originally have a biology background, and now I'm kind of in the middle of the two. And in particular, I really like understanding model behavior and why they make the predictions that they do. And then there's also other roles like product management, program management, and so on. And you'll see that the image generation put the ML engineering in the dark, coding in some sort of basement, which is accurate. I can attest to that. So yeah, in conclusion, I think AI in genomics has really transformative potential. It's the future of personalized medicine and healthcare. Our CEO, Demis Hassabis, said this quote once: "AI is the descriptive language of biology," which I think is really nice. I think it makes sense. And he won the Nobel Prize recently, so he knows what he's talking about. And just to summarize, it is the early days for AI in genomics. People are just now starting to build powerful models. If you're interested, it's a very good time to get involved in various ways. Thank you. That's all. Yeah, questions?
Hi, Natasha. Really great talk. I also belong to the intersection, like you said, and I have a lot of questions. I'm sorry for that in the start. So in your talk, you have talked a lot about ground truth missing, and that's the case in many fields. So when we have problems where there's no ground truth, you cannot see or evaluate how your model is, what your model is predicting is not, is it right, is it just gibberish, or what is it saying? What you are or the team is doing for that, because in AlphaFold's case, there were benchmarks to test it, and then there were X-ray crystallography models for the proteins, and they proved that it was really correct to some extent. So we have something backing that up, but antibody design and various other vaccine design, etc. fields, they don't have anything. We don't know if this model is true, if this antibody is true or not. And also, you talked about the RNA-seq being trained, a model being trained on that, but how do we discern that what the model is predicting, is that based on noise? Like these sequencing techniques, they introduce a lot of noise, and then we have various techniques in bioinformatics to get to the real variants and remove all the noise from the data. And since you say that there is no bottleneck for compute now, especially for the big companies, why do we not train a model that's trained on the coding DNA, then the RNA is transcribing, and the protein? So DNA data, its corresponding transcribed RNA data, and the translated protein data. Why has nobody built, just for the sake of seeing what it would do, train a model on that? Because we have just 22,000 plus proteins from the DNA, and that's not a lot. Just why has nobody done that to see what it would do?
Okay, whoa, that's a lot. So the first question is, what do you do if there's no data to train on or to evaluate on? How do you know if you're doing a good job? I'm not sure there's really an easy way around it. I think ML people are fundamentally reliant on there being experimental data. In some cases, you can do transfer learning, right? Like you train on something that does have data and it's slightly different, your application, but it kind of works. But in your examples, yeah, I mean, the data does have to be there fundamentally. What can we do? We're mere, computational people. And sorry, second question was
noise.
How do we know? Yeah, I mean, that's one that we have worked on a lot, actually. You might think that denoising the data before training on it would be better. You might think quality control filters on the data would be good. Yeah, we tried different combinations. I mean, I think deep learning is reasonably robust and it can learn to ignore things that are not relevant. There's limits to that. Yeah, I think it's an empirical question. It's a matter of exploration, how much you try and reduce noise or filter noise out. But so far, I've been surprised by how robust these models can be. And sorry, remind me the third question.
Training on the DNA and the transcribed RNA and the translated protein, since data is not that much, and we have compute.
Yeah, exactly. So actually, the bullet point that I put in, that's sort of a chili pepper, my hot take. I think to be involved in the field, you don't need massive compute, and a lot of the interesting work that I've seen recently is actually analyzing models that have already been built. But yeah, your question is, can we just train on coding regions? Sure, yeah, I don't see why not. People will do sweeps to people will try different regions to train on and see the effects of that. So that's a good idea. It's definitely, makes sense.
Yeah. I would hide the bullet point of bottlenecks by compute, because if we are doing our TPU for this year, you probably don't have show that.
Sure.
Happy New Year of Lunar Snake, Natasha.
Thank you.
And my question would be a little bit future and intuitive. Is there a possibility, and are there enough space for us to do gene editing in junk RNA in the future? I think this might be a direction.
Yeah, that's a great question. A while ago, there was this concept of lots of junk in the genome. It's true that only 2% of the genome codes for proteins, but that doesn't really mean the rest of it is junk. And more recent papers have shown that a lot of the genome is actually transcriptionally active and it's doing interesting stuff. Can we edit those regions in order to, I'm not sure I fully understood.
In order to cure diseases like bipolar or Parkinson's disease for human being, for Asian?
Yeah, no, that's a great question. I think people will continue to discover the importance of these previously called junk regions, and probably, yeah, they are causal in diseases like that. So, yeah, it's a very good point.
Thank you.
Thanks. You spoke about the importance of introducing benchmarks into a field like this. And I was wondering, is the sort of bottleneck for those benchmarks just the data, or is there also maybe some disagreement to what the actual metric that should be optimized is? Because as an example, in the weather community, we're not really bottlenecked by data, but people seem to disagree every single day needlessly on what exactly it is that we're aiming for in the field.
Yeah, absolutely. There's massive disagreement, actually. And I think it can impede research progress as well. Like we're writing a manuscript now, and we're making decisions about our evaluations in such a way that we don't have to defend it as much to people, so there's fewer arguments with the reviewers. I think if there was more of a consensus, or maybe some authoritative voice that just said, look, we are going to aggregate all these benchmarks into one suite, and that's it. But then there's that XKCD comic, right? Like, oh, these 13 previous standards are no good. We'll make one final standard. But then that just ends up being the 14th standard. Yeah, you're spot on. It's a big difficulty.
So I have a question, like if any research was done on like pairing these data with other ones, like for example, demographics or some CT scans or things that can then find a pattern or analysis?
Yeah, absolutely. One of the data sets I mentioned previously, you might be interested in UK Biobank. I think they've done the best job of this. I haven't looked too much into all of us. This is quite new, I think less than a year old. But yeah, Biobank will pair genomic information with like, health records, with readouts of all sorts of different experiments. And the task is, like, can you correlate these things in a meaningful way? So.
Okay, thank you. And there's another question also, like all the analysis for this thing is happening like after collecting the data, right? So there's no way to have it like in a real time analysis.
Ooh. What would that look like? Yeah, this is way not real time. This is way past data. What do you mean by, what would it be like to have real time?
Maybe like a real time can, so you talked about mutations and then how they happen. So they can happen like, you know, just any time. So let's assume that if the technology is getting like there advanced, that people can wear something that keeps monitoring their, like if there's a mutation or something. So that can happen in real time. Even it can recall the previous, let's say, sequence, and then what happened just suddenly.
Yeah, that's a very cool concept. I've never heard of real-time like monitoring of, I guess, somatic mutations, not inherited mutations. That would be cool. I guess it can happen anywhere in your body though. So how can you monitor like all your cells for like the tiny chance that a cancer pops up here or here? It's a very cool concept there, but I'm not aware of anything like that.
Oh, and no one is targeting such thing, right? Or maybe there could be, not yet?
I don't know. I haven't heard of it, but it's very cool.
Okay, thank you. Thank you so much.
All right, I think that's a final question. Yes, great. I think you have time for one more.
I'm sure you know the answer. I know very little about biology, but why is it a transformer the right mechanism for genomics? Because, you know, I guess a gene is unique, right? So, for example, here you're supposed to disambiguate words like the word bank. For example, it could be a river bank or it could be a money bank, right? And that's, so the embedding of the bank depends upon the context. Here, what is the relationship you're trying to, would the embed, because the gene only is unique, so it doesn't appear in many places, so its embedding would be the same, right? So maybe
Yeah.
I'm not asking the right question, I'm not sure. But
No, that makes perfect sense. Yeah, a gene is unique, but it's composed of many components that are not unique. So like I talked about transcription factors, which regulate the expression of many genes. And one transcription factor might have like 10,000 possible sites in the genome that it binds to. But that binding, the activity of that depends on context massively. So a gene's behavior is very context dependent. If its context is very repressive, it kind of doesn't matter that the gene is there, like it won't be expressed. Or if a gene, doesn't have a strong promoter, but 200,000 base pairs away, there's an enhancer that kind of counteracts that, then it will be expressed. So I guess it's exactly what you say, like for context-dependent cases with long-range dependencies, transformers do well. And that's exactly the case here, because lots of different sequences in the DNA kind of need to come together and interact to get gene expression.
so what's the word in this? Is the gene or the sub? What are tokens and words in this context?
Yeah, so most models don't do like explicit tokenization. We just model like the single bases. The model has to learn to define kind of its boundaries on its own. So we don't tell it that like, look, this is a binding site, this is a gene. The tokens are always DNA bases. It just develops an implicit understanding of these tokens, if you like.
Thank you very much.
Thank you.
Awesome.
Thanks, guys.
Thanks.